Amarachi, I still can't see the interpretation button on the ribbon.
I apologize. I don't know why. I can see it and all the languages are intact. Aspen, can you?
I cannot see it, Amarachi.
Oh, okay.
Okay. So English-speaking participants, can you see the interpretation icon on your Zoom ribbon, either at the bottom or the top? It will be a globe. Yes, we can see it. Great. So Amarachi, it seems that the participants can see the translation icon.
Yeah, they can all see it, but you are also supposed to see it. So I'm just checking the Zoom to see if there's some setting that maybe we did something there, but you can comment and then we will fix it as we go on.
It's okay. And it's now just ticked over to 2 p.m. 2 p.m. Geneva time, 1 p.m. West African time, and I guess it's 3 p.m. on East Africa. So I'd like to welcome everybody, whether it's good morning, good evening, or good afternoon. So welcome to this webinar from the WHO Regional Office for Africa on highly pathogenic avian influenza, H5N1. I'd like to welcome you all from wherever you're coming from. I know that this invitation has been shared widely, I think. So if you're joining from Europe, welcome. If you're joining from all the countries in Africa, good to see you back. I'm now going to introduce the speakers that we have today. Myself, Belinda Herring, and I'm the influenza focal point at the WHO Regional Office for Africa in Brazzaville, in Congo, and I work within the preparedness area of emergencies. We also have Dr. Jessie Bonwit, who is also within the Afro Emergency Preparedness Department. Jessie, would you like to come on and give yourself, well, introduce yourself, please?
Hello, everyone. My name is Jessie Bonvit, and I am the Technical Officer for Zoonotic Diseases and One Health. I am based at the Afro Emergency Hub in Nairobi, where I serve the Southern and Eastern African countries. Thank you, Belinda.
Thank you very much, Jessie, and welcome from the East African office. Thirdly, I'd like to introduce Dr. Aspen Hammond, who is sitting next to me in HQ. To introduce herself. She is from the Global Influenza Program here at headquarters. Over to you, Aspen. Excuse me.
Hello, good afternoon or good morning. And yes, I'm sitting in the room with Belinda, and it's a pleasure to participate in this webinar on avian influenza. I work in the Global Influenza Program on surveillance of seasonal and also zoonotic influenza.
Thank you very much, Aspen, and thank you for joining us for this Afro webinar. So next, I would just like to step through what we're going to have as the agenda. So we have the administrative tasks first, which I think we're a bit ahead of on time. Then we're going to, because it may be a very wide audience, a little background on influenza. So influenza 101. Then I'll give a brief update about the global and the regional situation of avian influenza. So that will be regionally speaking from the African region. Aspen will give us an overview of the current risk of the H5N1 and a specific clade of H5N1, which is called 2344B. And then I wanted to show some resources about where you can find information about avian influenza and situation updates and potentially things that you may want to find, like messaging for communities or things like that. We have a lot of time built in for questions. So we're going to have a brief question answer session if there's time after the global and regional update. And then another question session, brief question session after Aspen's talk on the H5N1 2344B session. And then we have questions at the end as well. So please feel free to ask questions. We love questions, and no question is a silly question. So please fire away. And that's what we're here for. I'm going to go over the housekeeping now. So this is a virtual webinar. It adheres to the Code of Conduct to prevent harassment, including sexual harassment at WHO events. There are some features on your Zoom ribbon, which you should be able to see. I'm not sure I can see them, but one should be, there's the chat, there's a raise hand, and there's a question and answer, and then the interpretation button, which is a globe. We would like you to put questions in the question and answer feature, and then Aspen and Jessie can answer them when I'm not speaking, and then I can answer them when I'm not speaking. Please put your questions in the Q&A. We'd prefer that to the chat. Also raise your hand. We would love to hear from you, and we'd love you to ask questions live. There is interpretation, so we will be speaking in English, and hopefully the interpreters will give me some sign if I'm talking too fast. The interpretation button is a globe on your Zoom ribbon, and if you click on that, you can choose which language. The interpretation services for this webinar are French and Portuguese and English. And please put a message in the chat if there is something that is not correct, or you're not hearing the interpretation, or you can't find the interpretation. You can have closed captions if you want, and that means that there is, I'm going to call it interpretive text of what I have been saying. So sometimes it gets it right, sometimes it has some funny words. So you can either have the captions on or have the captions off. And that is the little CC button on your Zoom ribbon down the bottom. Now, this session is being recorded, and we take your attendance as consent to be recorded. Recordings will be shared in the coming days following the sessions, along with the session materials. As we continue to improve our training programs, your feedback is invaluable to us. A survey will be shared at the end of the session. We highly encourage you to share your thoughts and suggestions. Your input will help us make our sessions better and also enable us to meet your expectations and needs. Okay, I'm going to stop there for a minute and ask if anybody has any problems with any of the interpretation or any questions to start off with. I know Amarachi has put in the chat that we would like you to introduce yourself, just briefly say your name and where you're from. I can't see any hands, but my trusty sidekick Aspen should be able to see a hand if you have any problems with interpretation. So I'll just pause here for a minute or so to see if anyone can. So someone's raised their hand. Use the thumbs up icon if everything's okay. So I see, Victor, you have raised your hand. Are you able to unmute? We've got lots of thumbs up. Awesome. Victor, I'm going to unmute you. And can you ask your question? Can you unmute? Am I right?
He needs to unmute from his end. I have given him access to speak.
Okay. Victor, please unmute yourself if you're going to speak. No?
I'm sorry, that was a mistake from my side.
Okay, no worries. Okay, so I can't see any other hands up. So I think we'll move on with an Influenza 101. So this will just be a brief introduction to influenza, the virus. And in this session, I'll predominantly be talking about human influenza. So influenza virus is an orthomyxovirus. It's a negative RNA virus and it has a segmented genome. And the virus is an envelope virus. And currently there are 4 types of influenza virus, 2 that are clinically important to humans, influenza A and influenza B. There is also influenza C, which undergoes antigenic drift, which I'll talk about in a little minute. And it only infects humans, and it only has 7 segments of its genome. Influenza D affects cattle and can spill over to other animals. This is a schematic diagram of an influenza virion, or a virus particle. What you can see here is the segments of the genome. So there are 8 segments in the influenza A and B genome. Each of these segments codes for a protein and is translated separately. On the outside of the virus, you can see a purple thing that looks like a flower, and then maybe a yellow thing that looks like a flower. So these are 2 molecules that sit on the outside of the influenza virion. One is hemagglutinin, and the other one is neuraminidase. So the hemagglutinin is a trimer that sits on the outside of the virus, and it helps the virus attach to cells and infect cells. The neuraminidase molecule is a tetramer, and it helps the virus move away from the cell after replication. And these are 2 important molecules when we talk about influenza, because influenza is subtyped according to the hemagglutinin type and the neuraminidase type. So you may have heard people talk about H5N1, H5N1. So what they're referring to is the type of hemagglutinin molecule and the type of neuraminidase molecule. If we look at how many types of hemagglutinin and neuraminidase molecules there are, there's around 18 hemagglutinin types and around 9 neuraminidase types. And this expands further as we move into the evolutionary space of influenza. If we look at the hemagglutinins, the molecules that predominantly infect humans are H1, H2, and H3. So that's depicted by the man and the woman there. These hemagglutinin types, or viruses with this hemagglutinin, can also infect pigs, so a little pig there, and they're also avian. So on the far right-hand side, there's a column of birds. So all influenza species infect migratory water birds. There are other hemagglutinin types which infect humans, but not commonly, and same with pigs. If we look at the neuraminidase types, only N1 and N2 commonly infect humans and also pigs. We can see there are other neuraminidase types that don't ordinarily infect humans, but they can infect other species. And once again, they all different neuraminidase types infect aquatic birds. For influenza B, there are only 2 strains. There's B Victoria and B Yamagata. And we think that B. yamagata may be is not circulating anymore following the COVID pandemic. Okay, some fun facts about human and seasonal influenza. It infects around 1 billion people per year. There's around 3 to 5 million cases that have severe illness from influenza, and from 290,000 to well over 500,000 deaths annually, and this is globally from influenza, human influenza. The common symptoms of influenza are having a runny, stuffy nose or potentially eye irritation, coughing, having a sore throat or difficulty breathing, having fever and chills, headache, body aches and muscle aches, and diarrhea. So a lot of these are typical symptoms of a viral infection. And as you can see from those symptoms, they're very similar to what you would see in, say, COVID, for example. The symptoms start 1 to 4 days after infection and usually last around a week. How we diagnose or potentially detect influenza is by clinical presentation. And what we call this is influenza-like illness. And so this encompasses a lot of those common symptoms. So having a fever, having a cough or sore throat are the 2 key symptoms that are looked for. We can diagnose or detect influenza infection in the laboratory or by using rapid tests. And PCR is one of the common ways of detecting influenza. And the sample that we would take is the same sample that we would take for COVID. So because they're both respiratory infections, and it would be a nasopharyngeal swab. Influenza is transmitted in the same ways that, say, COVID is, through contamination of surfaces, through aerosols, being close to people who have infection, and treatment is generally clinical. There are antivirals available. However, they need to be taken very soon after infection because of their mechanism of action. We also have a number of prevention methods. There is a seasonal influenza vaccine that is tailored for either the Northern Hemisphere or the Southern Hemisphere, which can be taken. And then the public health and social measures that we saw associated with COVID-19 infection of washing hands, wearing a mask, and social distancing. And I wanted to make the point that there are many other respiratory infections that can also present with what we call influenza-like illness. And these can include rhinovirus, the common cold, respiratory syncytial virus, SARS-CoV-2, and parainfluenza. But there are others as well. I mentioned I was going to talk about influenza and antigenic drift. So the reason why we have a new vaccine every year is because influenza virus changes. It is an RNA virus, and so it undergoes mutation each time that it replicates. So antigenic drift is essentially a drift away from the original. So every round of replication, the virus may include a different nucleotide, and these just accumulate over time. And so it's drifting away from what it originally looked like. So on the figure that you can see, on the left, there is a man who has been infected by a virus with all pink dots. And then by the time we get to the right, which may be a couple of years, it may be a couple of months, the virus looks a little different. And so antigenic drift is the reason why we have seasonal influenza outbreaks or epidemics every year, and why we make, or a new vaccine preparation is made every year. The other mechanism that influenza uses to change is called reassortment. And this is generally termed as antigenic shift. And so this is an abrupt change in those 2 molecules, so hemagglutinin and neuraminidase. So when a cell can be infected by 2 different influenza viruses. So in my little cartoon there, I've got influenza virus X with green triangles and green circles representing hemagglutinin and neuraminidase. And then the Y virus has pink circles and pink triangles representing hemagglutinin and neuraminidase. So if they both enter a cell at the same time, all those segments that translate the hemagglutinin and the neuraminidase can mix up, and then the resulting virus can look different from either the X virus or the Y virus, because there has been a combination of the hemagglutinin and the neuraminidase molecules. So you essentially get a new virus subtype, which there may be no pre-existing immunity in humans, and this may particularly be relevant if it's an avian influenza strain and a human influenza strain that infect the same cell. And this type of virus may lead to a pandemic because nobody has any immunity to this. And so that's why it's potentially called a pandemic virus, and why antigenic shift is so important with respect to influenza. I'm now going to quickly talk about animal or zoonotic influenza. So if we look at, and I will thank Aspen for this slide, it's very, very good. If we're talking about animal influenza, that is an influenza that circulates in animals. Remember I said there are lots of hemagglutinin types and lots of neuraminidase types. So there are influenza viruses that only infect animals. A zoonotic influenza is an animal influenza virus that then can infect humans. If we talk about a pandemic virus, that could be a pandemic virus is something that is generated either through accumulation of mutations or reassortment, and that is a virus that can spread among humans who have little or no immunity. Then we have seasonal influenza, which is basically generated by antigenic drift, and that causes seasonal epidemics. And so there is that need to monitor what is circulating at the moment so that we can identify if we have zoonotic influenza that may become a pandemic influenza or a pandemic influenza. Okay, we've talked about the fact that influenza can infect many animals. The natural reservoir is aquatic birds. And then I guess to follow on from that, there are a number of documented flyways for migratory birds, which are illustrated in the picture down to the right. And generally you see avian influenzas spreading along those particular flyways. And I'll talk a bit more about this when I say the regional update. So influenza has now been found in penguins, in seals, in cats, dogs, minks, bats, horses, and of course pigs. And then most recently, it's been found in cows. And Aspen will elaborate on this in a little while. So if we come back to this picture, we can see that, and I've only depicted humans, pigs, horses, and birds in this diagram, and then a seal. But we can see that there's a lot of other hemagglutinin types and neuraminidase types that have the potential to infect animals. So if I take an animal influenza as an example, as equine influenza virus, it originally spread from birds to horses. So the 2 subtypes that infect horses are commonly H5N1 or H5N1. And H5N1 is now probably the most prevalent one. H5N1 has been reported in other species. So as you can see from the image there, it's spread to dogs and it has established circulation in dogs. It has not been demonstrated as an acute infection in humans, but there is serological evidence that humans have been infected, and there's occasional spillover to camels and pigs. So this is an animal influenza. Now, the other important, this is a very important animal influenza, is swine influenza. So it's highly contagious in pig populations and causes significant morbidity. So the most common swine subtypes are H5N1, H5N2, and H3N2. And as you can see, it's the hemagglutinin 1, 2, and 3, which infect humans, and the neuraminidase 1 and 2, which infect humans. So essentially, swine flu can infect humans. The impact of swine flu with regards to pig populations is generally economic, with respect to the loss of weight for pigs going to market. And swine influenza infections aren't part of the WOHA, which is the World Organization for Animal Health. They're not mandated to be reported as part of WOHA. There is a public health risk with swine influenza, and human infections with swine influenza aren't common. But there are infections in people who have direct contact with infected pigs. The disease resembles seasonal influenza, and then swine influenza is rarely spread within a human population, and swine influenza is not a foodborne disease. So I'm going to try and speed up. So if we look at animal and how it would become zoonotic influenza, we have the swine influenzas of H1 and N3, and if they pass to humans, they are then called a zoonotic influenza. The same with, say, H5N1 in chickens. If it then passes to humans, then it would be called a zoonotic influenza. Okay, and this is just a diagram to illustrate how reassortment occurs, and this is looking at the H5N1 pandemic strain in 2009. So on the left-hand side, you can see a swine virus, an avian virus, and a human virus. All those segments mixed up to then form the two in the middle, which are yellow, red, and green. And then a Eurasian avian virus was then put in the mix, and so they all mixed up. And so on the pandemic strain has two Eurasian genes, has some North American genes, has an H3N2 gene, and has some swine influenza genes. And so this was a virus that was novel to humans, and there was humans were susceptible, and there was no immunity. So that just illustrates how reassortment can happen with the different segments of the virus. Infection with animal influenzas is not very common in humans. This is an epicurve of the number of infections of other hemagglutinin types in humans. And as you can see, there's not that many. H5N1 and H7N9 are the most common infections in humans. So just coming back to this and identifying the difference between animal influenza, zoonotic influenza, pandemic, and seasonal influenza. And I'm just going to give now a brief overview of what the regional and global situation is with avian influenza. So we're looking now at influenzas that infect birds and could potentially be transmitted to other animals. Okay, we're back here to our same picture again. And as you can see, H1 to 16 infect aquatic birds and N1 to N9. Okay, so avian influenza became, I guess, prominent or something of concern in the early 2000s, where there was an outbreak of H5N1 avian influenza in Hong Kong. And so it had a fairly high case fatality rate of around 30%, and all the strains were of avian origin. And at that point in time in Hong Kong, they closed all the live bird markets and they slaughtered chickens. So essentially, normally the response for an avian influenza virus is to mass cull chickens. In 2003 and 2004, H5N1 was reported in a large number of countries in Asia, and there was a lot of concern worldwide that it may be the next pandemic virus, which of course it wasn't, because there was the swine virus in 2009. But there was, an expanded range of the virus, Moving on, and there were the first human cases in Vietnam late in 2004. The transmission of H5N1 was obviously from bird to human, generally due to exposure to ill poultry or due to practices such as plucking or preparing birds, handling the fighting birds, playing with poultry, or consuming undercooked poultry, or in Vietnam, I think a delicacy is duck blood soup. There was evidence of transmission of H5N1 to animals. Some of the leopards and tigers in one of the zoos in Asia were fed raw poultry, and they contracted avian influenza from that. And then the human-to-human transmission at that stage was essentially limited to household contacts or intimate contact with people. So it wasn't spreading beyond that. It stayed as a zoonotic influenza in the limited number of human cases. These are the migratory flyways. This is just I guess an enlarged image. And so a lot of them come from Asia down through Africa and back. And some of them can cross over. Now, these are predicted ones. So birds can stray. That's all I can say. Birds they have their own, they can fly. Okay. The number of H5N1 infections reported in humans from 2004 is over 900 in over 20 countries. So, and as you can see on this picture, the only human cases in Africa have been reported in Egypt and also in Nigeria. And if I just flip back to the previous slide, we can see that one of these migratory pathways basically goes over both these areas. The red one goes over Egypt and comes over Niger and Chad into Nigeria. And then some of the other ones from Asia come down the coast of West Africa. Okay. So, and this is a map showing the number of avian influenza events worldwide since October 2022. As you can see, there are a lot in Europe. There's a lot in North America. And I think this is the first time that there have been a large number of events in South America. And as you can see from this image, there's a lot of infections going up the west coast of Chile. And I think the next slide or the one after, I'll talk a bit more about that. There was also a number of events both in Southern Africa and also in West Africa. This is the picture from the 1st of October 2023 to now. So that's 6 months, 6 to 8 months. So we see that there's still a lot in Europe. There's a couple of events in Southern Africa, not that many in Western Africa, and certainly a lot fewer in South America. But I think I'm very disappointed about this, but it's touched the Antarctic Peninsula, avian influenza, and we have some outbreaks in Australia, but they're not H5N1. So if we look at this distribution, this is just giving you an idea of the increase of infections. On the picture that is showing now is what's happened in January 2020. And if I do this, we can see that in 3 years, there has been a massive increase in the number of infections in animals globally, and particularly of H5N1. And I talked about South America, and the hotspots that we've seen on those slides are Europe, North America, and South America. In particular, in South America and down the coast of Chile and up that coast, there are a lot of infections in aquatic mammals, so in seals, in sea lions, in dolphins, and there have been a wide variety of species infected in North America and also in Europe. And this is with the virus 2344B. And I've sort of explained this before. It's like where we see avian influenza infections predominantly in Africa are down the west coast of Africa and also in Southern Africa. So this is the situation since 2017 in the region. There've been H5N1 outbreaks in a large number of countries, predominantly in West Africa. but certainly down the coast in southern Africa. We've seen H5N2 infections, H5N6, H5N8, with large outbreaks in South Africa and I know Zimbabwe, H7 and H7N6 most recently. There's only been one human case of H5N1 reported in Africa, and that's been in Nigeria. And this is the situation that was from the WOHA report. Actually, no, it's the FAO report from the 11th of July. So we have events in Burkina Faso and the north of Nigeria and events in South Africa of H5 and H7N6. And I think that comes to the end. Yes, so that comes to the end of the regional update and the introduction. So that was to take us to 12:30. So we might open it for questions, I think. So I'll stop sharing my screen. And are there any questions? You can put your questions in the chat, or please feel free to raise your hand. We have plenty of time today, so I'm quite happy about that. Let me see if I can see hands.
Hi, Belinda. There are 4 questions in the chat. Do you want me to read them out?
Yes, or if you can read them out and answer them, or if you need, happy to. If they're easy, I'll take them, and if they're hard ones, I'll give them to you. Over to you.
Right. The first question is from Ibrahim.
Yeah.
Why are we having a new vaccine while we know that the viruses is mutating instead of having boosters?
So that's a good question, actually. So for influenza, you may change, the strain changes. So, I guess you could call it a booster, but it's not necessarily the same antigen that you're putting into humans. I don't know. Do you have a better answer to that, Aspen? With respect to boosters and changing of the vaccine? So they don't change all the components. So an influenza vaccine it's either trivalent or tetravalent, meaning it has an influenza A component, or probably 2 influenza A components and an influenza B component. So not all 3 change all the time. It may just be one, influenza A component. It's not likely to be an influenza B component because there's only 2 subtypes. So it's not the whole virus, not the whole vaccine that is changed. It may just be components of the vaccine that are changed based on the circulation of the seasonal influenza viruses globally. So what happens is there are 2 meetings each year, one to decide on the formulation for the Northern Hemisphere vaccine, one to decide on the formulation for the Southern Hemisphere vaccine, because each hemisphere has a typical influenza season. It's generally in wintertime. So they look at what is circulating with respect to the different subtypes, but also the different genetic and antigenic makeup of those subtypes. Because even though we see the subtypes, there's a whole range of antigenic and genetic variability within each of those subtypes. So if they have a situation where the current vaccine is not going to give protection for what is circulating based on a sort of an antigenic change in the virus, then they will change that component of the vaccine. I hope that answered your question. But a good question. Okay, next.
Thank you. The next question, there's one question from Juan Martinez, which I don't fully understand. So maybe Juan Martinez, if you could rephrase the question, that would be very useful. And I'll move on to the next question while you write it. So another question is by Alfredo Bruno. Could specific primers for the detection of seasonal human influenza directed towards the H3 subtype recognize equine, canine, or other species H3 subtypes? Same question for H5 primers. So I think the question is, can you use if you have a primer for H3, can you apply it across all species?
And I'm a human virologist, so I'm gonna to be honest, I don't know if you can detect H3, say equine influenza with the same H3 primers as human influenza. I'm not sure if Askman can help me with this, but thinking about the fact that you would be looking at they would be in different labs. It's very seldom that H3N8 infections transfer to humans, predominantly because the receptors on the cells may be receptive to H3, but the N8 may not allow for the virus to replicate very well in humans because it's not adapted to humans. So I think that's a good question, and I might do some research and look at that, because within the human space, the H3s will evolve, and if you look at a phylogenetic tree, they will evolve within humans based on the receptors that they recognize. In the equine space, the same thing will happen. So originally they may have been 100% identical, but over time, each of the H3s may have diverged away from each other. And depending on where you make your primers to in the hemagglutinin gene, if it's a conserved region, possibly. If it's not a conserved region, then maybe you would need specific primers for each of the different species, even though they are still H3. And the same would go for H5, but we do have the countries that are doing sentinel surveillance for seasonal influenza have access to primers that do detect avian H5 influenzas, predominantly so that we can check if there is spillover, because spillover is going to be significant, because we know that there's a high case fatality rate with H5N1 in humans. And same with H7. There's H7N9 is a high case fatality rate. So there are primers available for labs that are doing surveillance for influenza for H7 avian viruses. Okay, there's 2 participants with their hands raised. Should we look at those? So let's take one of those. So Kazora. I think you'll be unmuted by Amarachi.
Okay. Don needs to unmute and speak.
So if you've raised your hand, try and unmute yourself and then speak.
No.
Okay, Dr. Darar, I can see that you're unmuted now.
Dr. Halima, I'm muted and I'm unmuted now.
Okay, go ahead with your question.
Thank you. Thank you for the nice presentation. My question is, what shall we do globally as we've seen the challenge of it? Currently is pandemic and we have a challenge in vaccine as the virus is changing now and then, but also there is this movement from one place to another globally, and which also shows as the one of the main of cause of this pandemic. What shall we do? And as you say, the sign and symptoms, they look like the COVID-19, and even they are spread somehow. Now, my
Yes.
question globally, what are we going to do? What shall we do currently?
Right, so that's actually a very complex question, so I'll try and answer it. So pandemic preparedness is something that each country has been doing as a continuum for I guess, many years. And the expectation was that an influenza virus would be causing the next pandemic. It certainly wasn't a coronavirus that was going to cause the next pandemic. So there is a group here at WHO that's called the pandemic, or there is a framework, a global framework called the Pandemic Influenza Preparedness Framework. And within that framework, WHO and partners help strengthen the surveillance capacities of countries, the detection capacities, regulatory capacities to get vaccines into countries, and risk communication and community protection practices. So there is ongoing work globally to help countries strengthen capacities to deal with a pandemic. And we've made many gains as a result of the COVID pandemic. So the COVID pandemic, even though it was a different virus, it certainly helped advance some of the pandemic preparedness. Now, along with this framework, and it's a partnership between industry and WHO, there is a certain number of influenza vaccines that will be made available to WHO at a reduced cost to distribute to countries. With regards to having a pandemic vaccine, because influenza is an infection that we know about now, and we have we have a vaccine every northern hemisphere and southern hemisphere season. Once there is a pandemic strain that has been identified, it will rapidly be shipped to vaccine manufacturers so they can then incorporate that into production of pandemic vaccines. So as we saw with COVID, this was a new virus and it was a new pathogen causing a pandemic. So the ramp up to virus vaccine production was quite long because the vaccine had to be essentially formulated and then manufactured. Whereas with influenza, we already have those methodologies there, and it's just basically you put in the pandemic antigen into the vaccine, and then the vaccine can be produced. So the build-up between pandemic declared and vaccine being deployed will be much faster than we saw for COVID. And there are a lot of mechanisms in place globally and also probably nationally the pandemic influenza preparedness framework helps build all of those to help countries be able to address a pandemic. So I would encourage you to look at go to the health topics page on the WHO page, there is, and you look for seasonal influenza. The WHO website isn't the greatest to navigate. There's a section that says groups and initiatives. So one is the Pandemic Influenza Preparedness Framework, and the other one is the Global Influenza Program. So if you want to know more, click on those because there are some very good videos which briefly explain it. They briefly explain how it works with WHO, with industry, and what the benefits are to the global good. Okay, over for now. Dr. Derar, are you still unmuted? No? Dr. Derar, I can see that you're unmuted. Hazora, are you unmuted? Okay, Jessie, let's read one more question from the chat, and then we might move on with Aspen's presentation.
Okay, one question by Benedicta Boama is how can you contain unrecognized and combined genomic subtypes when there's an outbreak and there's more migration seen globally?
How can you contain unrecognized and combined genomic subtypes?
In animals?
Are we talking about in animals or birds or avian?
I mean, by more migration, I assume human migration, because I don't think we're having more animal migration.
Well, you get the birds migrate each year. Let me ponder on that question.
I think it generally goes back to surveillance.
Surveillance, yeah. So essentially, you know, we have influenza surveillance in countries, particularly in the human context, and I'm sure there's surveillance in the animal context as well in countries to try to pick up these unusual events. And so if you have, and this happens across Asia, and we're trying to really get it strengthened in Africa, if you have sick poultry or sick birds, then you notify somebody. And I have to be honest, I'm going to turn to Jessie because this is where the One Health aspect comes in, and the linking of human health and animal health to ensure that there are good surveillance practices, and particularly for influenza, between the human health and the animal health, and also the environment, because you may end up with an event where there is a lot of migratory birds that come to a lake and they all die. And because birds die, it's not always influenza. There may be other diseases that cause birds to die, but certainly in the environmental sector, recognizing those events and then addressing them to make sure that they're not animal influenza. With respect to humans, we're hoping that, you know, if there is an unrecognizable or a subtype that doesn't cause an overt disease in humans, that it is going to be mild. But certainly if you have a cluster of people who have, you know, died of a respiratory illness, it's certainly something that needs to be investigated. Okay, I think with that,
I would just
yes,
on that, if in people, if you have detected an influenza virus that's unsubtypable, the action to take is to contact your regional office, your country office, contact WHO, and also arrange to have that specimen sent to a WHO collaborating center so that further characterization of that unsubtypable influenza virus can be done quickly. It may be that it's a seasonal virus or it's a known animal influenza virus, but it's very important to get that further testing done quickly.
yes, I concur. And there are mechanisms to do that in countries. So particularly from the human health, but if from the animal health side, you would reach out to perhaps the FAO, OIE representative in your local community to alert people that something has happened. Now, Aspen, am I sharing your slides?
Sure.
Yeah. Okay, we'll move on with Aspen's talk. So she's going to talk about this H5N1 virus 2344B that has rapidly expanded globally. And if we think back to the slide that I showed with all the infections in Europe, all the infections in North America, and the ones down in South America on the west coast of Peru and Chile, this is the virus and the clade of H5N1 that is causing those infections. And it's something that has become perhaps on the concern ladder, has stepped up a rung of the ladder. So let me share Aspen's slides. Okay. Are you seeing it in presentation mode?
Looks good on my side.
Okay.
Yeah, thanks. So you can go to the next one.
Okay. Yes, thumbs up.
Yeah. This is this presentation, Belinda's given a good introduction to actually avian influenza, and I'm just going to go a bit more detail into H5 and specifically the clade 2344B H5 viruses, and focusing a bit on the event of the detection of H5N1 in dairy cattle in the United States and WHO's risk assessment. So this is a nice infographic from CDC that gives a bit of the background again, as Belinda talked about the different times in the past where H5N1 viruses have emerged in animals and causing occasional infections in humans. Next slide. So and the next slide just shows a bit of the the virological information. So looking at this this lineage of Eurasian H5 viruses and how they've evolved over time. So again, there was an emergence in 1996, and these viruses, as they emerge and change, as Belinda said, they fall into sort of groups, and we give those groups numbers because it's easy to describe these viruses that are similar to each other using these numbers, these clades. And so in the middle, you can see this is a tree that lays out different viruses and shows how they fall into these different clades. So in that between 1996 when these viruses emerged and now, there were about 10 clades, but in recent times it's really only the clade 2 viruses which have persisted, and among those, clades 2.3.2 and 2.3.44 and 2.2 have persisted. And since 2016, it's really there's really the 2.3.2 and the 2.3.4.4, which have continued to diversify and form multiple subclades. So you can see that in the picture on the top right, the colors indicate the clades over time. So we can see there's a lot of purple, which is the 2.3.4.4b viruses, which are predominant globally in the past few years. And these viruses also evolve into different subtypes. As Belinda said, sometimes there's reassortment. So the H5 viruses may combine with other viruses. So you get a new subtype like an H5N6 or an H5N8. And those also circulate in birds and occasionally have infected people as well. So it's a complicated virological picture, and there's a lot to monitor over time. Next. And so what I wanted to go further into a bit more further detail is the geographic spread, especially since 2020 over the past few years. So the map on the left shows 2020, the H5N1 viruses as they sort of originated in Asia, spread to Europe, Africa, also spread to the Americas, North America, and have continued to spread to South America. This has happened to varying degrees and since the virus evolved. But again, this is just focusing on this most recent intercontinental transmission. And another concerning aspect is the increase in animal species that have been affected over the past few years. So on the right of the slide, there's two images. One is from 2003 to 2019, showing the species of animals infected with H5N1. And on the very right is a comparison from the years 2020 to 2023, showing quite a larger variety of species infected. This can be due to a lot of different factors, the spread of the virus to different locations, different animals are exposed. It also is related to surveillance. So there's a lot more surveillance happening in different animal species. So it's due to many factors that we see this spread into different species of animal. Next. So as I said, we've seen in 2021 H5 spread from Europe into North America, and this was concerning. H5 viruses had previously spread into the Americas, but this happened again in 2021, and these viruses continued to spread down from North America into South America. Next. The other, as I said, the concerning issue is that we've seen H5N1 outbreaks in different species of animals. And over the past few years, we've seen not just detections, but outbreaks in mammals. So there was an outbreak in 2022 of H5N1 in farmed mink in Spain. There was also an outbreak of H5N1 detected in domestic cats in Poland. That was a year ago. Also a year ago, there were domestic cats affected in South Korea, in the Republic of Korea. And also last summer, there was a large outbreak of H5N1 in farmed fur animals in Finland. In these events, it's difficult to say whether there was mammal-to-mammal transmission. It seems likely in some of these events, but it's very difficult to have definitive evidence to say that this is the case. And on the bottom, again, just this highlights the spread of H5N1 viruses down the coast of South America in migratory birds and birds, but also in marine mammals, sea lions, seals, going down around the tip of South America and then back up the coast on the east side, causing massive die-offs of birds and mammals. And we're in the situation in this year where we have a new event, which is the detection of H5N1 in multiple livestock species in the United States. So initially there was a detection in goats in a farm, and these goats had very close exposure to infected birds. But there was a separate event where H5N1 was detected in dairy cattle as well. Next. So Belinda showed already an epidemic curve of human cases of infection with avian influenza viruses. I'm highlighting the past 6 or so years, so since 2018. So we see there's a continued detection of zoonotic virus infections in people. There's a wide variety of influenza subtypes, though. So it's not just H5N1 we see, but it's a lot of other subtypes. We have H10 viruses infecting people, swine influenza viruses, H6, and different H5 subtypes, as well as H9N2 viruses, which are very commonly detected in birds in many regions. So we're talking about H5N1, but I just want to highlight that it's important to be vigilant that other subtypes can infect persons, and we need to be aware of that when we're doing surveillance. Next. So just to summarize a bit, some of the trends in the global spread. So we've seen around 900 cases, as Belinda said, of H5N1 in humans since 2003. Specifically the H5N1 viruses of the clade 2344B continue to diversify genetically and spread geographically, and also infect a broader range of species, so wild birds and mammals, causing significant mortality events in animals. Despite this high number of outbreaks in animals and the large exposure of humans at this interface, the human-animal interface, there have been relatively few human infections detected and reported to date. Importantly, among these, no sustained human-to-human transmission has been reported. And this is an important point. This is something that's very important in the investigation and the assessment of events when a person is infected with an animal influenza virus. We always need to be vigilant and be looking out for any unusual events that may signal human-to-human transmission, because as we heard, that could be the signal of the change to the virus, that the virus acquires the capacity to spread from human to human, and that could signal the beginning of a pandemic. So again, these viruses are constantly changing, and we need surveillance to be of quality so that we can make sure we're detecting unusual events and keeping an eye on viruses that are circulating. And again, with surveillance, timely outbreak investigations and reporting as well. Next. So I just will turn, I think, for the few slides coming to the event of the H5N1 infecting dairy cattle in the United States. A bit of a timeline of this event. So it's thought that in late last year, early 2024, this unexplained illness in dairy cattle in the United States was associated with H5N1 virus. So there were dairy cattle that were experiencing a drop in their milk production with other non-specific symptoms in multiple states. Eventually, after some investigations and testing, H5N1 virus was identified in the milk of some affected cows. And this was reported from USA to the World Organization for Animal Health at the end of March. at the beginning of April, a first human case of infection following exposure to infected cows was reported to WHO, and this person had conjunctivitis, so redness in the eyes. Their subsequent important points in this event were that there was detection of virus particles in pasteurized milk. And so there's been a lot of studies looking at the effect of pasteurization and the safety of dairy products. A second human case was detected in May. Also somebody who had exposure to infected dairy cattle. This person also had conjunctivitis. A third case was reported, and this person had exposure to infected dairy cattle, but also had respiratory symptoms, not just conjunctivitis like the previous cases. And where we're at now is there was a fourth case reported in a person who also had exposure to infected cows in June and developed conjunctivitis. And as of a few days ago, I think the numbers already changed because this is a rapidly evolving situation, but there's been over 150 detections, 150 dairy cattle herds in 13 states affected, according to the information coming from the USA. Other aspects of this event is that it has been seen that the H5N1 virus that's infecting cattle has spilled over to domestic animals that are near these dairy cattle herds. So cats that are on these farms and wild animals such as raccoons and I think opossums, but also birds have been infected with the virus that was transmitted from the infected cows. And then this this week, the beginning of this week, WHO was notified of 4 cases of H5 infection in persons, in people who were working at a poultry depopulation operation at a commercial farm in the United States. They had really, direct contact with infected birds, and these birds were infected with the H5N1 virus, the same virus that is infecting the cattle. So it's a rapidly evolving situation. Thus far, again, the human cases that have been reported to WHO have evidence of direct exposure to infected animals. Next slide. So as I said, the agriculture authorities in the US are testing a lot of samples and are releasing this information on their website. So you can see this is the epicurve of the dairy cattle that are testing positive, the dairy herds that are testing positive over time by state. And you can see a map to date of the affected states in the country where dairy cattle have tested positive for H5N1. Next. And then just to summarize the human cases. So again, we've seen several cases with poultry exposure, and we've seen several cases with exposure to infected dairy cattle. Again, these cases have had mild illness. There's been thus far no documented onward transmission from these cases to their contacts. But again, the most recent situation is still under investigation, where the poultry depopulation was occurring. That was just a recent event, so still more to come on that. Next. And so there's a lot of questions, around this event. This is a fairly, it was an unexpected event. Dairy cows were not expected to be susceptible to influenza and spread it to other cows and spread it to humans. So there's still a lot of questions, and I think this is this is another good infographic from CDC showing different routes of transmission. It's suspected that the initial route of transmission was from a wild bird to a cow, and that happened again probably sometime the end of last year or the beginning of this year. But now we know that there's ongoing cow to cow transmission. There's also cow to poultry transmission. And also transmission from cows to other domestic animals and wild animals, and also cow to human. And so far, for human to human transmission, none has been reported to date. Next. And so the next few slides are just a really high-level overview of some of the latest research findings. So as this situation is very new, there's a lot of observational studies and also experimental studies that are starting, and publications are starting to come out with a bit more information on this event. But essentially, the virus, the H5N1 virus in dairy cattle seems to be replicating in high levels in the mammary glands of the affected cows, resulting in a high viral load in the milk of infected cows. There seems to be less replication in the respiratory tract, so there's very few nasal swabs from infected cows that test positive. It seems there's also a role of asymptomatic cows testing positive and potentially have a role in spreading the virus to other cows. And again, there's still a lot of research on different routes of transmission and spread. Next. So again, I think this highlights the what I kind of covered about different ways that the virus might be transmitted. It's suspected that there's probably likely ways that the virus is transmitted from cow to cow, but definitely cow to cow movement between farms is contributing to the spread. Cow to cow movement within a farm is involved in transmission. Fomites, so trucks, workers, milking equipment, all those probably have a role in spreading the virus between cows and between farms. Next. And this back to the viruses. This just highlights the diversity of H5N1 viruses in North America on the left, and in the right we can see that the H5N1 viruses that are infecting cattle form this tight group of viruses, all very similar to each other. And this has been designated as specific genotype, the B3.13 genotype. So all the viruses that have been detected from cows to date fall in this very similar group, as well as the viruses, the few viruses that have been characterized from human infections as well, all fall in this specific genotype. And so there have been some studies looking at the duration of shedding in cows. So it seems that cows are affected maybe approximately for about 2 weeks, having a decrease in their milk production that might last longer than that, but having some clinical disease for around 2 weeks. There's evidence of seroconversion in infected cows. And again, there's this role of subclinically or asymptomatic cows in the spread. So then this slide just gives a bit more information and the link. We won't go into detail, but if you're interested later, then that information is available there. Next. And some of the other interesting things that are starting to come out are looking at the distribution of the influenza virus receptors in cows. So we know that that's important in looking at species that are susceptible to infection and the routes of infection and the clinical signs that they get. So it seems that in the mammary glands of cows, they do have the receptors that allow avian influenza viruses to bind and replicate. So this is some of the recent findings that are coming out and give it a little bit more evidence to why the virus is excreted in the milk of infected cows. Next. There was also recently a study of experimental infections of cows with these viruses. So a lot of experimental studies are done with mice and ferrets, which are fairly small animals, but cows is a different story. So these studies have taken a while to get going because of the logistics involved in having this animal in an experimental situation. But a study was done with infecting 2 lactating cows and also 4 non-lactating cows to demonstrate that that inoculating the virus into the mammary glands did result in infection, and also that inoculating the virus intranasally into cows resulted in infection, but with very few systemic signs following intranasal infection. Next. Yeah, thanks.
Anticipation.
So moving a bit towards the risk assessment, what WHO does with a lot of partners in the animal health sectors and with our WHO collaborating centers is look at the characteristics of the viruses from animals, but also those that are infecting humans. And from this event, the conclusion is that thus far these viruses are primarily avian viruses, not adapted to replication and sustained transmission in humans. So there's been no changes that would say that there has been a change in the transmission, making these viruses more likely to transmit from human to human. And so the genetic analysis is an important part of this, as well as the antigenic analysis. So for the genetic analysis, look at markers for adaptation to different host species, studies in animals, looking at transmission, immunity, and the antigenic analysis for WHO is important to look at the pandemic preparedness, and if there was to be a need for a vaccine to be made, that we are ready and we have selected the vaccine so that producing the vaccine can be done more quickly and efficiently if there was a need. And so quickly after these viruses were first identified, WHO went through the process of looking at these viruses and looking at how they are related to the existing candidate vaccine viruses. And the existing candidate vaccine viruses that have been selected cover well these new viruses. So gives a bit of reassurance on that side. So in summary, the H5N1 cases reported thus far, these cases have direct or indirect exposure to infected animals or contaminated environments. So that's the likely source of exposure to the viruses. Infections in people can be severe and even fatal. The most recent cases we've seen in the US have been mild, but we know in other infections in other countries, infections can be in humans can cause severe disease and have fatal outcomes. So this is a serious event to be considered, and this is why surveillance is very important, so we can detect human cases quickly and get the treatment that the infected persons may need quickly. Again, there's been no evidence to date of further spread from human cases to their contacts. There have been no cases detected in the community where these cases have been detected. If there are cases detected in the same community, they've also had exposure to infected animals or their environment. The sequences from the virus isolates from the human cases are similar to the viruses that are circulating in local animals. But again, the prompt and thorough investigation of human infections are critical. And this includes, again, looking for any signals, any type of events that might indicate that there's a change in transmission, that the virus might be spreading from human to human. So that's why contact tracing and monitoring exposed persons is so important. Next. And I'll finish just highlighting the risk assessment. So following the notification of this event in March in dairy cattle and the detection of an associated human case, WHO with animal health partners at FAO and WAWA did a joint risk assessment looking at the public health risk and deemed the public health risk overall to the general population to be low. Again, these viruses have not yet acquired the capacity for transmission to human. We expect that further cases might happen in people who have exposure to infected birds or animals or environments, but in people without such exposure, the likelihood of cases is low. People obviously with occupational exposure might be at a higher risk of infection, and this is why it's so important that we emphasize that workers should have access and be trained to using personal protective equipment. Next. And again, as I mentioned, WHO also looked at the viruses and their relatedness to the candidate vaccine viruses. So there's more information on this slide with the links, etc. if you want to look at that after the webinar. I'll end there, and I hope that that was interesting, and we can have some questions later.
I saw some hands go up. Thank you very much, Aspen. That was terrific, and I saw some hands go up while you were talking. So let me figure out how to get to the hands. Participants. So I see one hand is up from Ali Kabir. So you've been unmuted. So if you could ask your question.
Good afternoon, Ma. Can you hear me?
Yes, we can.
Yes. My question is, you see, what are the risk factors that will catalyze the speed of antigenic drift and antigenic shift? And how are we going to mitigate it? That's my question mark.
Okay, so antigenic drift is something that happens during viral replication. So the only way to mitigate that is to prevent infection in humans. So genetic drift will always happen. Genetic shift is something that we can try and mitigate, but it's also difficult. So that may be that you have to limit the interactions between migratory birds, poultry, pigs, and humans. And, in some cases that's unrealistic to think about, particularly because, you can protect. So say, for example, you're looking at migratory birds and poultry, having your poultry housed so they're not, there's no access for migratory birds, things like that can help mitigate that risk. But it's very difficult with influenza to do this type of mitigation. So, I guess one of the key things is rapid detection using surveillance, and that's both on the animal side and the human side, and the environment. It's really important to help rapidly control an outbreak and help it and stop it from spreading. Anything to add, Aspen?
No, I think that's it's difficult because as we said, the natural reservoir for influenza viruses is wild birds, and it's there's not much we can do to control the virus in the natural reservoir. So in terms of the larger pool of influenza viruses, I think that they'll continue to evolve in animals.
Jessie, are there any questions in the chat?
We don't have any new ones in the Q&A, although what has come up a couple of times in the Q&A and in the chat are whether there is any experience in detecting influenza viruses from animal samples by using CDC test kits. So I know that, Belinda, you were going to look into that, but this has come up a couple of times.
So from what I understand, the CDC test kits can detect H7 infections and can detect H5 infections. I did see in the chat that the CDC primers don't detect H3 variant infections in swine. But there are primer sets that can detect H5 and H7 infections that are available through IRR for people who are within the WHO scope of influenza surveillance. But I think particularly with H7 and H5, there may be a biosafety limitation on who has access to those primers, but I would need to check that with the International Reagent Resource, purely because they are considered pandemic viruses or viruses with pandemic potential. So they want to make sure that people who potentially do have a specimen like that have the right biosafety requirements in their laboratory to be able to test it without exposing the workers, and also for escape of the virus, potential escape. If there's no other questions, we might move on to the, hang on.
Maybe, I think there's a question from Rodrigo. So the hypothesis of transmission of the virus within cattle, As far as I know, I don't think that there's a definitive answer for this yet. I think there's different routes that are under consideration. It seems that that one possibility is through the mechanical transfer. So when cows are milked, you know, a person is there. There's also a milking machine, so that the virus might be on hands or other parts of the milking apparatus and be spread from cow to cow. And the initial study shows that the virus can enter the mammary gland and have an ascending infection into the mammary gland and cause an infection that way. It also seems that infection via the respiratory tract is a possibility. But I think there's still a lot more to be understood on how the virus is transmitting from cow to cow. It seems that there's a lot of movement of lactating cows from state to state, although there have been, you know, response measures to this event to implement testing before movement. But it, you know, I think it still might happen that infected cows may be moved either, you know, they aren't showing symptoms before they're tested, or they develop, you know, infection just after they tested negative. So it looks like it continues to affect new dairy cattle herds and new areas in the United States, but there's still more to be learned on exactly how it's transmitting from cow to cow.
Thanks, Aspen. And I see an absolute expert is on our call, Richard Webby, Dr. Richard Webby.
Oh, no.
Who's one of our one of the WHO at the WHO Collaborating Centre and does a lot of work on avian influenza. So, Richard, please feel free to pop in if you have any pearls of wisdom on any of the answers of the questions. The next part of this webinar was to sort of go over some tools and resources of where you can find information on avian influenza or the highly pathogenic avian influenza. So I think if there's no questions or no more questions to move with at the moment, we might do that. I left plenty of time for this to roll out today. So are you seeing my screen in presentation mode? Yes. Okay, so this part of the presentation, and this is not exhaustive, this section. There are lots of resources from various countries on avian influenza. So remember, avian influenza has highly pathogenic H5N1 has been in Asia for a long time. So there's, you know, there's resources from Cambodia and Indonesia and places like that. So don't look at this as the only places that you can go. So obviously there's WHO, which deals with the human health side of things, but there's also FAO and OFLU, which is a collaboration between FAO and the Organisation Mondiale de la Santé Animale, which used to be called OIE, but it's now WOAH, W-O-A-H. So I'm just going to go through some of the things with respect to these four organizations. So, and I'll just give a self-plug. We had an avian influenza meeting in April this year with 11 countries from the region to look at how human health goes about investigating avian influenza outbreaks and to try to help strengthen that surveillance, say during the investigation or post the investigation, to look at potential spillover in humans. So some of the key takeaways from that was a checklist for investigation, which Aspen and I worked on, and Jessie, so that you have sort of a checklist to say, if your veterinary colleagues say we've had a report of, you know, 600 birds dead around Lake Albert, or that when you can go and investigate with the veterinary personnel and also investigate, say, the towns nearby and the people who are the occupational exposure to see if there's spillover. There's also a consolidated list of tools that are available for you. And then at the meeting, we asked countries what they thought their top 5 gaps were for preparedness for this type of event. And one thing was risk questions. So, and this will become sort of, I guess, more important if there is the potential, say, for example, of human-to-human transmission in the US. What is your risk question going to be for the potential of a pandemic virus coming to your country? Also having messages for communities ready to be rolled out quickly if there is an event in your community, in your environment, that you can roll out messages to say, you know, if you feel sick, go to the doctor. Don't touch or eat sick poultry. So that's just some of those things. We're happy to share the checklist with you if you need it. This is a brief example of a couple of pages of the checklist, looking for, you know, what you would need to have in the laboratory for testing, what you would need for surveillance. And, you know, I think a key one there for surveillance is looking at the list of triggers for investigation. So what would make you as on the human health side go and investigate something, having SOPs. So we're very happy to share that. It hasn't been formalized yet in any way, but that may come. I wanted to also highlight the tool for influenza pandemic risk assessment, which is a risk assessment of animal and zoonotic influenzas that WHO does to look at. I'm going to move past that slide because you can all read it. It looks at what is the risk of sustained human-to-human transmission of a particular virus. So it's done per virus. There's a number of risk questions that assess 2 components: the likelihood of the virus acquiring the capacity for sustained human-to-human transmission, and what would be the impact if there was sustained human-to-human transmission. So this is something that is done with WHO and a number of partners and experts globally. And they have done one, or they did one previously a couple of years ago for H5N1, say 2344B. However, as we've seen since then, there has been an explosion of cases, in which case there's an explosion of information. So, you know, another risk assessment will be done probably with this virus. This is a multi-step process. As I said, it involves partners and experts globally. They gather information and data on that particular virus. So it's done on a virus generically as a global entity. The risk elements that are looked at are human infection, disease severity, population immunity. They look at the geographical distribution in animals, the infection in animals, whether the virus can bind to receptors of human origin, the transmission in animal models, susceptibility to antiviral treatment, and genomic characteristics. So these risk elements have different weightings, and a final score is determined. The scores are averaged within each dimension, and you end up with something like this. So each of these risk elements are here. The coloured circles, so you can see that some are darker and some are lighter. That is a measure of how confident the expert panel is in the information. So for some dimensions or risk elements, there may not be that much information around, and then for others, there may be a lot. So the confidence in the information is also considered. So what you end up is something like this. So on the y-axis is looking at the impact, and on the x-axis is looking at the likelihood. So it's a likelihood of sustained human-to-human transmission and the impact that would have. So for example, I'm going to take this 2016 H5N1. So when the expert panel looked at it, the virus from 2016, it had a fairly moderate likelihood that there could be sustained human-to-human transmission, and the impact was fairly high. So the impact is around about 8, so fairly high. Whereas if we look at the avian H3N8, and remember H3N8s can also infect horses, the likelihood of transmission was low and the impact would be low. So where does 2344B sit in this? Which is the virus that we've been talking about. If we look at the 2021 clade and then 2016 and 2018 of H5N6, we can see that in 2021, the 2344B clade had a fairly moderate impact, but a moderate probably to low likelihood of transmission. And I think the low, moderate, and high, they've changed on the axes because the high should be around 10. So moderate's going to be around 5. So there was a fairly low to moderate likelihood of transmission, and the impact was probably low to moderate. Whereas looking at the 2023 virus, and this isn't this analysis isn't complete yet, the likelihood has increased a little, but the impact has also increased more than the likelihood. So this is something that is done at WHO with partners and experts globally. It's done on a regular basis on different zoonotic viruses. There is also a joint risk assessment, and Jessie can perhaps speak more on this if there are questions. It's a risk assessment, say on potentially avian influenza transmission or avian influenza, and it's done in collaboration with FAO, OIE, WHO, and the environmental sector. Some countries have done joint risk assessments for various diseases. This is also a multi-step process, and there is a tool for an operational tool for countries to do a JRA. And so this is freely available on the internet. You can download it and have a look at that joint risk assessment. WHO also has a number of resources. In the top left there is the landing page. If you type in, so you can see on the top row there, there's something that says health topics. So if you go to health topics and you type in influenza and avian and zoonotic, you will land on that page there. And there are a number of resources on that page. Just doing a quick scan of the internet, I found some frequently asked questions from the Southeast Asia region, which you could have a look at. There's also these 2 key documents, the summary of key information, practical countries for experience outbreaks of avian influenza, and that is in English and in French. And then on the far right is the public health resource pack for countries. So within that publication, all the resources on avian influenza are stratified by different topics, say for risk communication, for investigation, and it gives you the web links or the URLs. If you're looking at it online, it's the web links of where to get that information. So there's the WHO webpage. There's a couple of key publications. From FAO, they have a situation update. They have a web page which gives you the situation update on different avian influenzas, on SARS-CoV-2, on MERS. So if you look in the bottom right-hand corner there, they actually have a sub-Saharan Africa highly pathogenic avian influenza update. So you can click on that. They also have an introduction to avian influenza, a self-paced course. So if you wanted to find out more, you could log on and do that course. And these are just brief snapshots of information that is available. Obviously, OIE or WOHA has much more information. They have information about the dynamics of avian influenza, how to build policy for avian influenza, preventing it, protecting your poultry, which was just a question that we had before, whether you enclose them, whether you vaccinate them. And they, as a result of the outbreaks in South America, they published this guide for responders to outbreaks in marine mammals. The WOHA also have situation reports on avian influenza. And so if you click on one of the situation reports, at the very end of it, it has the links to all these different resources that you can access and have a look at, videos. There's the OFLU information. There's links to the risk assessments for the joint risk assessment, the cumulative numbers of cases for WHO, what's been recent in the news. So there's a lot of information out there. You can get regular updates from WOHA about animal diseases. So that's something you can also sign up to. And I think that's it from me. Jessie, did you want to say a couple of words about the joint risk assessment, maybe, and how that would work with avian influenza? And then we'll open it again for questions. And maybe if Richard, you're still on, you might want to make a comment about the risk at the moment. So over to you, Jessie, first.
Sure. So the risk assessment is a tool that was developed by the tripartite, so WHO, FAO, and the World Organization for Animal Health. And it's essentially a formal mechanism that allows you to do a risk assessment jointly between multiple sectors. So usually animal health, human health, and wildlife health, and sometimes also the environmental sector. So it follows the classic procedure of a joint risk assessment. So in that sense, it's nothing new or that you don't know if you have been conducting joint risk assessment. But the guidelines that you can find online, they just walk you through how to do it in a structured way that includes all of the sectors.
Okay, thanks, Jessie. And I will just add that when I was looking for resources, I did find a nice podcast by Richard. So if you type in, sorry, I hate to plug your name, Richard, but Richard Webby, you'll come up with the podcast. Richard, would you like to make any comments? And I'll ask Amarachi to unmute you. Richard,
Yeah, can you hear me okay?
Yes, we can.
And hello, everybody. Yeah, so I think looking at the risk, particularly in the context of the bovine outbreaks, the risk to human health at least does seem to still be very low. There isn't any evidence yet that this virus has substantially changed upon passage from cow to cow. So it's still a virus that is mostly, well, almost fully avian in all of its properties. Its receptor binding are still very much avian-like. Its replication kinetics are very similar to 2,3,4,4-Bs that are circulating in birds. So, the risk from the virus itself is still very low, still very much an avian virus. But of course, the more opportunities we provide this virus to interact with the human host and infect people and potentially evolve, clearly that's not a good thing. So there is I would say there's a slightly elevated risk just because of the fact there are probably more people being exposed to high levels of the virus. I don't know if that was useful, Belinda, but happy to
No,
add
I think it's always useful to hear from an expert, Richard. So thank you very much. And I don't see, is there one hand up? No, maybe not. Anna Woiso. Anna, are you unmuted yet or? No, I'm asking so it says you can talk. So if you unmute yourself, Anna, and ask your question, that would be great. Can you find the unmute button? No. Okay, so I see that from Aspen, we'll wait to see if Anna can unmute. There's a lot of questions about primers for H5N1. So as I said, the labs that are part of the Global Influenza Network, which do surveillance for influenza in countries, do have access to H5N1 primers that detect human influenza. From the animal side, I would have to investigate with my One Health colleagues because they would be in touch with the FAO or OAE or WAHO people who would be providing a supply of primers to the veterinary labs. But certainly if you suspect an H5N1 case in a human, then there are primers available. However, with that, if you have somebody who presents with an influenza-like illness, the first point of call is testing them to see if they are influenza positive. Remember that influenza-like illness is a syndromic condition, and so it can be caused by bacteria, it can be caused by viruses, and from the viruses it can be, caused by influenza, SARS-CoV-2, RSV, rhinovirus. So then once you have identified it as influenza, you would then type it to say if it's influenza A or influenza B. And then if it's influenza A, you would subtype it with the common garden varieties of subtypes that circulate globally. And then as Aspen pointed out, if you weren't able to subtype it with those primers, you would then ring the alarm bells and send it to a WHO collaborating centre. You would contact your WHO focal point to say that you had identified an influenza A that was unsubtypable. And then the processes, the wheels would go into motion to get that sent to a collaborating centre so it could be characterised fairly rapidly. So testing a specimen or testing a person that comes in with influenza-like illness, you would not be testing those for H5N1 in the first instance. And if in the remote possibility, because we've identified that the risk is low, that there may be, and I'm saying may, be human to human for H5 transmission, then obviously primers like for COVID would be readily available. So I hope that's answered that question. I don't know that there are any more questions. I'm just going to turn to my colleague Aspen. No? Jessie, any more questions?
I think that we've covered a lot.
Okay.
Don't see any.
If there's any remaining questions, please feel free to either contact myself through perhaps your country office colleagues or your influenza lab. Once again, there is lots of guidance on avian influenza globally, maybe not in cows, but certainly in birds. Access the WOHA site, FAO, WHO, whatever you need to. And I think with that, I will thank Jessie for moderating the question answers. I'd like to thank Aspen, my co-presenter. I'd like to thank the team that organised this, our team in Nairobi. So big claps for you guys. And thank you very much. And thank you to all the participants who attended. And of course, if you have any questions, please don't hesitate to contact us. So have a lovely afternoon and a great weekend. Bye.
Thank you for the translator.
Oh, yes, sorry, I forgot the important translators. So thank you very much. Big claps to you too. Thanks so much and see you next time. Bye.
Bye.